In this video, we will discuss about this matrisome recapitulation in ex vivo models. So far, we have discussed that these PA-ECM ex vivo models provide the characterization of the pancreatic cancer cells that is similar to the pancreatic cancer cells that are observed in the patients that are affected with the pancreatic cancer cells, pancreatic cancer disease. Or we can say that the characterizations of these ex vivo models by the PA-ECM is more close to the in vivo characterizations that are observed compared to the other ex vivo models. So in this video, we will discuss about their approaches or their attempt to reduce those differences that they have observed in the ex vivo models and the in vivo models. So in order to reduce the differences, they culture the pancreatic cancer cells in these different ex vivo models along with these macrophages and pancreatic stem cells, so that they will get the characterizations more similar to the in vivo observations, as in vivo conditions also have pancreatic stem cells and macrophages. So what they have found out is that in terms of the interstitial matrix protein, the number of proteins found in the case of uh, primary tumors are higher, or in the in vivo models are higher compared to the ex vivo compared to the ex vivo models, whereas in the case of basement proteins, the basement membrane proteins expressions, they are higher in the case of ex vivo models compared to the in vivo models. And the reasons they have mentioned for these differences is that the in vivo process is a long-term process and it takes times, and during those times some of the ECM gets sacrifices and might goes inside the cells, whereas in the case of the ex vivo models, they are the short-term process, and the and it does not basement membrane proteins does not get the opportunity to go internalize within the cells. That's the reasons the basement membrane proteins are higher in the case of ex vivo models compared to the in vivo models. And these things are also validated using these collagen expressions, where we can see that collagen 3 expressions are higher in the case of ex vivo models compared to the in vivo models, where in the case of in vivo models the collagen 1 expressions are higher. And in the literatures, past literatures, it has been mentioned that during the time of this tumor onset, the collagen 3s are expressed initially, and afterwards the collagen 1s are expressed. So these things also substantiate their previous finding that the because of the short term process in the ex vivo models, they are able to see differences in terms of these matrix proteins that are expressed in those samples. However, one more thing that the researchers has pointed out is that the compositions of the PA-ECM resembles more to the primary tumor cells compared to the organoid. And after that, the next important proteins such as laminin, they also check the laminin compositions in the different ex vivo models and the primary models. And what they have find out is that the PA-ECMs will provide the compositions that is more similar to that of the primary tumors, whereas the organoid has more number of where the organized compositions is more different, or has a higher difference compared to the case of primary tumors expressions of the laminin. So from this they are able to find out what are the reasons why their ex vivo model shows difference compared to the in vivo models or the primary tumors, because of the time that in vivo models takes for the tumor development compared to the case of the short times that are exposed by the tumor cells in the ex vivo models. And the other compositions expressions such as collagen 3 expressions and collagen 1 expressions compositions are also more closely in line with the primary tumors compared to the other ex vivo models such as organoid. And things are also been validated by laminin compositions also. So all this indicates that their PA-ECM models is much better models much better ex vivo models for the characterization of the pancreatic cancer cells compared to the other models that have previously been used for this pancreatic cancer cell culture and their studies. If you need to understand more about this experiment in detail, you can go through the reference that has been provided in the description. And these data are being analyzed based on the mass spec analysis of these samples. So that's all for this video. Thank you for your time.